Welcome everyone to our penultimate session in our CCMA SRL perspective series, looking at high parameter or integrating multiparameter cytometry into your SRL. We have a really great panel today. It should be an excellent discussion. And before we get started, I just want to thank our sponsors, BD, our series sponsor, BioLegend, Cytek, and Sony for sponsoring this session. And with that, I will hand it over to Emily.
Awesome, thanks, Asia. Hi, everybody. Thank you so much for joining us today for, as Asia said, our penultimate session. We do hope you'll join for our last one in a month or so too, and we'll be advertising that at the end. So yeah, today we're going to talk about integrating multi-parameter or high parameter cytometry into your facility. Or, ooh, okay, if you guys could mute. Okay, great. Integrating multi-parameter or high parameter cytometry into your SRL or your core facility. So as I just said, we have an awesome panel today. I think they really need no introduction if you guys are in the flow cytometry world, but I am going to ask each of our panelists to give a quick introduction to themselves, where they are and their background. So maybe Pratip, you're top of my screen there. Would you like to kick off?
Sure. So my name is Pratip Chattopadhyay. I'm currently the founder and CEO of Talon Biomarkers, which is a CRO that's focused on high parameter immune monitoring. And I'm also the chief scientific officer of a new company in the data analysis space called TerraFlow that we're spinning out of NYU. I used to be an associate professor and founding director of the Precision Immunology Laboratory at NYU, and before that, I trained with Mair and was a staff scientist at the NIH in the immunotechnology section for quite some time. And before that, I was an infant. So yeah, that's my story. And I guess we'll hear from Brian next.
Hi, thanks. I apologize. COVID happens, and I have finally got the supply chain showed up this morning, and they're just delivering. So might be a bit AFK for a bit. My name is Ryan Brinkman. I'm a professor at UBC, and I work. My day job is at the BC Cancer Institute. I've been doing flow cytometry informatics for about 15 years, I guess. Spun off a little company. My main focus is helping people with their analysis by developing and applying automated methods.
Awesome, thanks Ryan. Thanks for joining us. Florian.
Hello everyone, my name is Florian Mair. I'm currently a staff scientist at the Fred Hutchinson Cancer Research Center in Seattle. I've been doing basically cytometry and single cell analysis for the past 10 years. Both the other speakers here, Pratip, Ryan, have been like some of my idols in the past, so I learned a lot from them, and I'm currently exploring data analysis, trying to bridge this kind of gap between wet lab scientists and bioinformaticians and trying to help people do good science.
Awesome, thanks Florian, and welcome. And last but certainly not least, Cindy.
Hi everybody, thanks for having me today. So maybe I'll start out by saying that I think I started doing flow cytometry before any of you were born. So, but I'm not going to tell you how old I am. I did my first flow cytometry experiment actually as a PhD student at the University of Alberta. So there you go, long, long time ago. Then became more interested in it when I was a postdoc at Stanford in Irv Weissman's lab, and I worked very closely with Mario and Gary Nolan and even Alan Stahl back in the day. purified my own APC. So I think those are my bona fides. Anyways, since coming to Toronto for my faculty position, where I've been ever since, I'm a professor of immunology at University of Toronto and a senior scientist in developmental stem cell biology at SickKids Research Institute. And I have directed the flow cytometry facility at SickKids, and we're actually interinstitutional with UHN for a number of years. And about 6 years ago, 5-6 years ago, we sort of launched an offshoot of the flow facility that we now call CASCA, or Center for Advanced Single Cell Analysis. And that's where we rolled out our CyTOF-based platforms. And we've also recently added the BD Rhapsody single cell RNA-seq platform. But we'll probably incorporate the higher parameter flow on that side as well. Anyways, enough said about me.
Awesome, thanks, Cindy. All right, so to get us started, I was thinking we could first begin with chatting about, what actually is multi-parameter cytometry, or what qualifies as high parameter cytometry these days, because as we know, technology has evolved a lot from our exciting 4-color experiment on a 1-laser flow cytometer back in the day up to many, many parameters now. So what do we call high parameter? Where, if you're in your SRL and you think you're doing high parameter, how do you know? Is it 10 plus, 15 plus, 20 plus, or are we talking about up to 40 plus parameters right now? So would like to hear your thoughts on that and where we are at present. So I don't know who would like to start.
I was saying earlier, it sounds a lot like a discussion that was just on the Purdue list, and I think there's one nugget that came out of Mario who said polychromatic might be a good way to talk about it. I think a bit of it's like next gen sequencing, and it just gets more and more and more. I guess high parameter is whatever we have today, because I don't know, because what's the next step after high parameter? I mean, it's like what was that band? We go to everyone goes to 10, but we're going to go to 11. Right? Where can you go after high? There's nowhere to go. Higher.
Yeah, it's funny when we were doing 12 color experiments, 15 color experiments, and then we started doing 18 color, and then we went up to 28. It was always, what do you call it? Do you call it polychromatic, or is it, and in various grants or various things where I've given talks or places like that, I've called it variously polychromatic, high definition, ultrachromatic, and every time you're struck by how clever you are for coming up with a new terminology. But ultimately, I guess one question is, does it really matter? We're all doing more parameters, and the thing that's heartening to me is that it's becoming more common. Cytometry is really becoming about high parameter work more and more, and that's really I think a good thing for the field.
Yeah, maybe I'll just weigh in. I mean, I agree with what you guys have said, that in some ways, what does the name matter? And, the field, of course, is always going to move towards more and more parameters, higher resolution, deeper profiling. But I think the other perspective I would offer, and maybe Emily wants to comment on this because she spends a lot of time in my facility helping people implement various kinds of panels. And what I've seen in my own lab, and I think a lot of our clients, is that we have a lot of people, like we have, 200 plus users of our core. We have a lot of people who are routinely doing, I would say, less than 6 parameters or less than 8 parameters. And and for a lot of them, they have very defined questions. They just need a few markers. That's fine. And where the challenge comes is, I would say, moving above 6, 8, somewhere in that perspective, just because the experimental design and the computation just gets much more challenging, and certainly requires a lot more input from experts like Emily and core managers who can help people really make that transition. So in my mind, as you move beyond some of the older standard fluorochromes into the newer fluorochromes and higher parameter, It just gets a lot more complicated to plan and execute properly. So to me, that's a bit of a dividing line.
Yeah, that's actually, sorry.
No, so go ahead, Florian.
Yeah, that's actually, I mean, a nice point where I can continue. I mean, I think the terminology high parameter always depends on where you're coming from, and based on that, you'll make your definition. But my very pragmatic definition is high parameter starts where you can't just throw stuff together and it'll work. So basically, like you can throw together a 10-color panel and it'll work. And beyond that, you need to start thinking a little bit. And to me, the two important aspects is that doing any type of high parameter work now is just much more standard. And we used to have only classical fluorochrome-based cytometry, and now we have just a variety of different ways how we can go about that. We can do classical fluorescence-based cytometry, we can do mass cytometry, we can do sequencing-based technologies together with single sequencing-based surface protein phenotyping, together with single-cell RNA sequencing, which is of course a different ballpark, but it's still the same approach and the same underlying technology. And I think that's where things get really exciting. So yeah, it's a matter of perspective. And for me, it's the moment you need to start thinking about what you're doing, that's where high parameter starts and gets exciting, I think.
So I'm going to hop in here and tag off of that. So you mentioned that there's a number of different technologies, techniques, and it's not just limited to like our classic conventional flow cytometry. How do labs go about choosing which technologies to implement in their facilities, which ones to consider, which ones may not work for them? And how do you start parsing through these decisions? Because not only are the experiments expensive, but so is the technology. And a lot of labs can't afford the entire suite of every possible technology.
You know, so one thing that I think we have to start realizing is that these technologies are means to an end, right? And that the end is usually similar, right? We're trying to better study biology. I have seen institutions and and organizations collect these technologies, which strikes me as really a waste of money. I think it's important to have maybe a couple of options available, but for goodness sakes, do you really need to have mass cytometry and MIBI and imaging cytometry and imaging cytof and and also have high parameter flow and have and that there are certainly reasons to have each and reasons to have more than one, but I think we're reaching a point with the technology development where there are a lot of analogous technologies, and I think it'd be a better use of institutional funds to choose thoughtfully rather than to purchase every one. Investigators will always want to have their own unique one, right? Because it makes them different. They have their own interests and biases, and it becomes incumbent upon the people at cores to smartly guide those conversations.
Can I just jump in here with one thought? And that is, I think you're totally right with what you're saying. And I think any of these technologies is only as good as the people operating it. And I think that's where the investment needs to be and where the education needs to be, because you can produce garbage data with any of these technologies. Some, of course, are more prone than others, but anyhow, and if you have a skilled operator or like a well-educated operator, let's put it that way, or experimenter, then you're going to get good data. And I think it's an important task of the community actually to have good educational resources and have strong data that highlights the strengths of certain technologies versus the weaknesses, so that people can make educated decisions versus just someone saying, oh, I think that's the coolest instrument on the planet because it's pink or yellow or purple.
Yeah.
So I agree with everything that's been said, but I think I'm going to provide a couple of different perspectives. One from perhaps a client perspective and one more from a core perspective. So, I think in a perfect world, if a new client or collaborator comes to our facility and says, okay, here's my project, here are my questions, what technology should I use? We would direct them in the appropriate direction based on what we had available, obviously. But that almost never happens, with an exception I'll talk about in a minute. Mostly what happens is people come and they say, well, I'm continuing a project that was started by a graduate student or another postdoc or a technologist, and they use these markers, and I want to do the same thing, but maybe I want to add to, or I want to use some new fluorochromes that are out there or whatever. So that's a whole different thing, because if you were starting from scratch with that person, you might say, well, you should just go straight to our, 28 color cytometer, or you should go straight to CyTOF. But they never want to do that. And often labs don't want to invest, right? Because they already have a suite of reagents and they don't want to start over. They just want to kind of incrementally add. And often, you can do that, but it's not ideal, as if you were starting from scratch. So that's one perspective. The other, though, would be, and I think the exception, or an exception to the example I just gave, would be, we're often approached being at a hospital-based research institute by clinician scientists who really don't have a history of doing this kind of work. And they may never have done flow cytometry or anything like that, but they have a patient cohort and question, and they want it, so they really are starting new. And so for those sorts of clients, we would say, well, you should just do CyTOF, because it's easier to, cast the net wide. You don't have a lot of cells, you don't have mice that you can keep going back to, and you want to use as many markers as possible. And, and we try to make it easier for those kinds of clients and collaborators, because we have facility panel that they can get started with to get pilot data and then decide if they're going to expand. So those are the different perspectives that I guess I would add to that question.
And I just want to, that's great points. And I just want to sort of add on to this, and reading some of the comments on here about, you get the instrument and you don't have the appropriate people. Part of it is just driven, I think most of the people on this call are academics. Part of it's just driven by the nature of academia. And, sometimes, I know in Canada, certainly it's much easier to get money for the next shiniest new piece of equipment.
Absolutely.
Than it is to get smart people.
Yep.
And so the challenge is we can write the grants to get, the next CyTOF or the next whatever, but it's really challenging for us as PIs to write the grants to get people. And that it's sad. And I don't know if it's like that in the States as well, but the machines are sexy and people aren't. And it's much harder to write the research, to get the right people. You have to write the research programs, and those grants are much harder to get funded than writing the grants to get instruments. And that is to the detriment of science, unfortunately.
And if we think about the numbers, I mean, a new instrument in this space is often $1 million, right? How far could $1 million go to staffing up, or supporting the careers of people who might be on the cusp of losing funding or something like that? And so there's some real thinking from a big picture, from the system's big picture, that we have to consider here.
Yeah, absolutely. I think that's true. I think we, CCMA, have hosted a session previously talking about grants and so on. And, this was definitely a topic that came up, and Vera has touched on it in the chat too, about maybe something we should bring up with the funding agencies, instruments versus people, and which is the most important. I think, just kind of, speaking about what's exciting and what's new and what's sexy, as Ryan said, something that's very in vogue right now is, everybody wants to do it, is single cell RNA-seq, which falls into this kind of category too. So I was wondering, kind of, could chat about, if you're a cytometry facility and you want to think about incorporating that into your facility, how do you kind of go about that? What are the important things to think about? Fostering relationships with your genomics core and so on. And also, if you have any thoughts on the workflow, what does the facility need to provide versus what the user should actually be doing as well. So would like to hear your thoughts on that.
So Pratip, you want to start or do you want me to start?
I'll let you go. I have lots to say about this, but
I'm sure you do, and I'm sure Florian does too. But
Yeah.
since maybe I'm the newcomer to this area, maybe I'll just give you how we've done it. You know, and my interest in expanding into this technology came partly from my own science, because you know, for many of us, that's where the science is taking us, but also from again being a scientific director of a core for a long time and having people come to us and they want our help in prepping cells for single cell RNA-seq analysis, you know, mostly using the 10x system, but also having conversations with some of the leaders of our genomics core. We have at SickKids, I think it's the biggest genomics core in Canada and probably one of the largest in the world, but they don't deal with cells. They you know, and they were always sending people to us saying, these people want to do single cell, we have the equipment, they just need to bring us the perfect single cells and we'll run it, but we're not going to help them prep the cells, we're not going to help them, you know, do anything. So, you know, after hearing this for a number of years and realizing I needed to move my science in that direction, I thought, okay, well, we have to get into the space. And luckily the Rhapsody was just coming on board, and I, you know, heard some early work about the platform, thought it had some really attractive features, and thought that we could actually provide support for it as a flow core. And so that's what we've done. You know, the equipment itself is not that expensive. It's having the highly trained people that, of course, are the ongoing cost. So the way we work it now, and this may evolve, but we will help clients with all of the upstream prep. So we will help them identify optimal tissue digestion conditions that don't destroy epitopes. We will help them do cleanup, sort if necessary. So we try to take them through all the steps up to putting them in the single cell capture system. Then we do that. We do labeling as needed with multiplexing antibodies or oligotagged antibodies for phenotyping, capture the cells. We take it all the way up to the cDNA, and then we hand it off to our genomics core. We could make the libraries, and we have done that, but I just thought it wasn't really good use of our time when they are the experts. And so we coordinate with them. They make the libraries, do the sequencing, send the data back either directly to the client, or if the client wants, we will help with the initial stages of the analysis. So we've been doing this for, I guess, about 1.5 years now, and I would say it's going well, and there's a lot of interest. But it you know, it does require pretty much a full-time dedicated person. So.
Yeah, I think that has been a great summary, and I mean, I couldn't agree more. I guess my perspective is in that sense also unique, that I was lucky in the past 5 years that I got to do these experiments really start to finish, meaning from getting the human tissue until all the way to the multiplex libraries, and I've done that both with the 10x platform and with the Rhapsody platform. And only once you've done the entire workflow, you appreciate the complexities of it, and why it's just doomed to fail if you have a core sorting cells, handing them off to a genomics core that's never seen cells, doesn't know how to count cells, which is not their fault because they've never done it. And then the finger pointing starts, where the core says it's the fault of the operator says it's the fault of them centrifuging the cells, and so on and so forth. And I think it's a huge waste of resources, waste of energy. And I think that's where we as a field need to tremendously improve, to appreciate that now is the time where genomics and flow cores, I'm not saying they need to merge, but they need to work much more closely together, or at least have one person that really knows the entire process from start to finish, and knows how to troubleshoot. Because as said, you can really, take the Rhapsody. The Rhapsody has this great feature that you can run the experiment, and then you have the cDNA on the beats. So if you mess up afterwards, you can go back and restart your experiment, which is actually fantastic, which is not the case for 10x. But so you have a chance to restart, but for a 10x experiment, you can't. So you really have to make sure that it works in one go. And if you do finger pointing afterwards, you're not going to solve it. You really need to know what's happening along the path, and just do it right, which boils down to education and like working closely together.
Yeah, and you know, the hubris that each group brings to these experiments is really important, and putting aside that pride important. You know, I noticed, so I was on the original CiteSeq paper where I did the flow cytometry benchmarking work, and the developers of CiteSeq were, I mean, they were just fantastic. They're super, super collaborative people, but they felt that they could sort of do everything, and they ultimately did, right? But the problem was that then when they started to expand to more antibodies, they couldn't tell if the antibodies were working because they didn't have experience looking at flow data, you know, and so they'd come to me, which was great for me. And similarly, I had this hubris that, you know, oh, I can do all of this myself. I don't have to worry about the molecular piece. You know, I'll just get the cells and run the whole thing. And, you know, and I felt like I was just dropped in an ocean when it came to trying to figure out how to sequence, how to figure out the depth of sequencing that's needed here, you know, how many reads, how to calculate that stuff, how to do all these. I mean, these are things that we are completely unfamiliar with, that genomic scores, it's like the back of their hand, you know? And so it's important to have kind of both these knowledge bases for the work, and to find people who are willing to be partners, you know, rather than, you know, think that they can do it all. I've seen genomic scores that felt that they didn't need the help of anyone who knew flow cytometry because, you know, after all, they had the 10x instrument and they had a protocol, and so that's that, right? And that's naive. That's a naive way to look at it. So
these things also need to be reported, though. I think this is what the field is not doing, because it's easier for everyone if you can look it up. What sequencing depth did these people use, and what data did they get?
Yeah. And you know, and the other thing about this is that these technologies are so expensive, and you know, and so you're really only getting, you know, one shot at this. And so if you know, if you don't sequence with enough depth, if you've, you know, not set up your experiment right, if you haven't tested the antibodies and they're not performing properly, then, you know, then you're cooked and you've spent a whole lot of money for essentially nothing. These technologies, by the way, it's worth noting that that they are incredibly powerful for the number of parameters that you're getting, but right now, at least, and this is probably going to change soon, you know, you're typically doing it on a small number of cells, much smaller than what we do by flow cytometry. And so doing, you know, rare events on bulk populations is difficult. You know, figuring out how to design experiments in this space, you know, you have to just be thoughtful about it, because you can't run too many cells with these technologies.
That is a really good point. It seems a lot of the success of these big multi-parameter experiments really comes down to proper experimental design from the outset. Are there any tips that you would give to users, flow core managers, on how they can, I don't know, work with this with their users? Anything that you've learned over your career that's been really fundamental in your success in these technologies and systems?
I mean, I have a quick thought. Please go ahead, Cynthia.
No, it's okay. I was just going to say, I think, you know, as you start to use more and more parameters, whatever the technology, usually it means you're interested in rarer and rarer subsets, or subsets that are more and more difficult to find because they need more markers. And so one of the things we've really started to emphasize in our training, and anytime we sit down with new clients or collaborators to talk about a high parameter experiment, is you have to start with the end in mind. What do you know about the rarest cell type you're interested in studying? Or do you not even, like, you have to have some idea in mind, because it boils down to, as Pratip said, you know, numbers of cells to analyze. So if you think you're looking for a 0.01% population, maybe single cell RNA-seq isn't the way to start. You know, and you have to think about enrichments and all those sorts of things. So I think really having a good handle on what you think you're looking for, while also being prepared to find things you weren't looking for. But you need to have those conversations in the beginning. And again, as I mentioned in the beginning, I think often people are just, you know, I understand that they're working day to day and one step to another, and they don't sort of step back and say, okay, what am I really trying to do here? And am I collecting enough cells? do I have enough parameters? Do I need to pre-enrich in some way to get reasonable statistics? All those kinds of things. Those are really key.
Yeah, the thought that I just wanted to bring up is kind of circles back to what I said in the beginning, that I think planning is much more crucial. And as a core, I think what you can do is really try to get these like few key papers in the field that we have by now that have reference data sets, and also these new journals that are now focusing on methods, which you might think is super boring. Like, for example, CellPrice has now these STAR protocols where we actually published a Rhapsody protocol. But if you would have asked me 5 years ago, I would have considered that boring. But now, knowing how important that is, I really think that it's critical to gather some of these reference data sets and some of these reference protocols and really go through them step by step, maybe even reach out to the authors, or I mean, do it yourself, obviously, in the end. But before you get started, really plan it well. And for example, coming back to this rare cell topic that has been mentioned now, what we ended up doing and what has worked really well for us, because we wanted to have bulk and the rare cells. For example, for many of our experiments, we sorted CD45 live cells and then had the markers for whatever rare subset we had and just enriched it into that bulk population on top of it. So being able to do both the bulk and have enough cells of the rare cell subset. And for this, for example, if you want to do like CytSeq, AppSeq, whatever, you need to think about how you're able to co-stain as antibodies, for which luckily there are now also protocols in this STAR protocols database and in other places. So I think it's just much more planning and much more need to study the literature and what other people have done and learn from it. I think that's key to success.
One thing that Florian got me thinking of is a word of advice to core leaders. These protocols are really detailed and there are many steps involved. It's way more complicated than add the antibody and wash. And so, when you're going through the whole protocol, and the STAR protocols, by the way, that Florian's group wrote are just, that's an awesome protocol. One of the things that I did here as I was trying to do the experiment, rather than walking around with a raggedy sheet of paper that's all stained up and checking off each step of 80 steps as I'm going through and doing it, was to actually rewrite the protocol in a format where the calculations are done on a Google Sheet or something like that, and put it up on the Rhapsody or on the CyTOMETRY's monitor, so that everything was electronic and my record of doing every step was electronic. And I think either journals or labs would certainly cores would be doing a great service to sort of generate these for the really complicated protocols that we're doing in this space.
Yeah, absolutely. I think that's a really good point. Just circling back to, excuse me, Cindy's point about, beginning with the end in mind. It's really important, and, thinking about statistics and all these kind of things. Just to chat to you guys a little bit about data analysis. Some papers came out, great papers, but scary papers about reproducibility and so on in the last couple of years. And one of the things that's highlighted, apart from, all your protocols and all that stuff, is that data analysis can be a really big source of variability and lack of reproducibility, especially in the cytometry world, with manual gating and so on. As we move into the more high parameter stuff, we're looking at more computational analysis and trying to remove some of that bias. Would you guys like to talk a little bit about that and where we're at and where we need to be going, and how experts do we need to be, and then we need to provide those experts in the facility, or is that something our users should be learning themselves in the background?
I think we're at the stage that it's possible to transition a lot from developing these tools to applying them. I think the evidence is pretty compelling that they can do as good a job or better than manual analysis. I can understand a lot of hesitation, and that I think a lot of that comes from it's scary and it's new and a lack of understanding. But I think if people take the time to delve into it, I think it's a compelling argument to adopt these tools that are available. Using the right tool in the right place is a challenging thing, especially when there's so many tools available, and education's got to be a very big part of that.
Yeah, so I think there's a lot to unpack there. So I'll try to just hit a few things that come to mind and let other people comment. But yeah, I mean, certainly as you get into higher and higher parameters, you just can't do the traditional manual analysis that I personally love to do. I like to see the data. I like to play with it. It speaks to me. But anyway, so and I agree with Ryan that there are some great tools that are fairly easy to use for people like me who don't have computational skills, and those are a great place to start. And I think we should be teaching our core facility clients who are working with higher parameters. We should be educating them to use some of these basic and easily accessible tools. But I don't think that should take the place of at least some manual exploration of the data. And often people start, even if it's high parameter, they'll start with a focus question on a particular cell type. And the best way to do that initially is to do your old-fashioned manual gating. But I think what I've seen sometimes is that I've been on graduate student committees where they've shown heat maps of marker expression on cells and the heat maps normalized in some way. And then when you ask them to actually show you the raw data, like there was no signal, but you can Z-score anything. And, it looked like there was a big difference and there was no expression at all. So, you have to use common sense, and sometimes that's not easy to convey or teach. And I think minimally, before investing a lot of time in doing clustering or whatever algorithmic approach you want to use, you have to make sure that the data is good. And usually that means looking at it manually. What's the range of expression of the markers? Do you have some markers that didn't work as well as they should? Do you have background that you don't expect? All these, and these are things you learn with experience and are time consuming, but otherwise it's garbage in, garbage out. So that's my comment there.
Yeah, it's really great that you bring that up. I mean, we actually recently wrote a piece about that. You can basically Z-score everything. And that is, I think it is a real problem. And I mean, Ryan left now. I think 5 years ago, there was this prospect of computational analysis making everything more reproducible. And of course, that still is the case. However, right now, I have the feeling that we are a little bit falling into that hole as a field where people just throw out the t-SNE heat map or like whatever normalized heat map and then interpret biology off that without keeping in mind that the underlying data needs to be solid, be it out of not knowing or be it out of pure ignorance. That's the part that I think is sometimes not entirely clear. But I feel that's where the whole reproducibility aspect of bioinformatics is falling short, because you need to circle back to the raw data and the methodology. And I actually don't have a good solution for that, but I think that is the real challenge that we have in the cytometry field right now, to make sure that all these fantastic tools we have at our disposal are utilized on good data in the right manner to interpret biology and not interpret artifacts. I think that's a major topic.
So, I totally agree. In a lot of ways, I just want to yell amen. One of the things though that the field seems to be moving toward, that is, really become standard for publication in a lot of places, is that you have to validate the results. You have to have a training and a test set, or, some additional experiment that is proving that that the signal that you're coming for, that you're reporting from a t-SNE is confirmed. And that's the right direction to head in. And I would encourage core leaders in particular to be aware of that, to encourage your users to design experiments in such a way that they, have a confirmatory validation cohort or set of samples that they can, confirm the result in. And that all points to the importance of bringing people who are good at the bioinformatics in early on in the study, bringing people like Ryan into the fold when you're designing the studies, rather than, sending them your crappy data after it's been collected.
Yeah, for sure. I think that's I guess that's a sort of a good topic too. Like, how does core facility, we talked about, having the people is really important. Getting a bioinformatics person just to do that in the core is probably going to be pretty pushing the core's resources. What do you guys think about that? Like, who should we be collaborating with, or where to kind of find those expertise? You have any thoughts on that?
Well, I think we have to probably distinguish different subsets of bioinformatics expertise or kinds of bioinformatics expertise. So I think mostly what we've talked about today so far is the kinds of things Ryan has been so key in helping us all use and develop and appreciate. But as he said, there are tools now that can be applied, and there will always be a need to develop new tools. But my experience recently is that certainly at the graduate student level, more and more of them are just learning R as a matter of course. Like, it's not so unusual anymore. In fact, most of my graduate students speak R, even though I don't. So that's kind of almost a basic level of competency that probably needs somebody in the facility to have, because then that opens up a whole bunch of tools that are sort of plug and play to some degree. So for clustering and classification of cells, there are other ways, and I think you mentioned this at the outset, Emily. So so Flojo has plugins that allow you to use some of these methods in a pretty straightforward manner. People like to use Cytobank. We like to use Cytobank because there are clustering algorithms that are really straightforward to use, and the data is all archived in a way that's traceable, which is nice. So those kinds of things I think should start to just become part of our language, and that I think ideally all facilities should be able to provide that. Although depending on the size of the facility, I recognize how challenging that can be. When it comes to the bioinformatics that's more sequence-based, and the single cell RNA seq analysis, there we typically rely more on I'll call them traditional bioinformatics, genomics bioinformatics types, and again, many genomic cores will offer sort of pipelines that they'll run your data through, and if that's all you have, I think that's fine. But personally, I really like to empower myself and the people I work with to do as much of the analysis as they can, and to, again, there are tools that allow you to do that kind of analysis without the expertise. And I think the power is in having the person who knows the biology really be the one to interrogate the gene lists and decide on cutoffs and all those sorts of things. Because if you have somebody who's a dry lab person who doesn't understand the biology, they'll make decisions. But I think somebody who does understand the biology will just bring a more fine-tuned eye to making those decisions. So.
Yeah, again, I mean, this is really something that I briefly want to echo, because for what you said for the single cell RNA-seq analysis part, because these data sets are so rich that if you have a professional bioinformatician analyze it, so if you have this kind of gap between a bioinformatician and a biologist, what I feel happens is the biologist does the experiment, then it gets sequenced, the bioinformatician does like a standard analysis, you get heat maps, clusters, whatnot, then it goes back, and then you look at the data as a biologist and say, oh, this is interesting, I want to look more at that, and then you might write a request to the bioinformatician, and like 1 week or 2 weeks later, you get something else. So it takes forever, and you're not fully exploring the data, and this is why at least from my perspective, as I've been so lucky to analyze my own data, this is an iterative exploratory process where if you want to best leverage single cell RNA-seq data, really you need to analyze it as from a biological perspective, because it's kind of, you ask something, you look at the molecule, you look at subsets, you look at heat maps, you find a new gene, you Google the new gene, you look at existing data, okay, that has been done, etc., etc. You can't really do that if these are separate worlds. I feel you really need to kind of bring them together in order to be most efficient with gaining insight that is relevant for revealing new biology. At least that's my take on it.
Yeah, for sure. Pratip, do you have anything to add?
Yeah, I mean, I was just thinking that we have a vast ecosystem of software that helps us with our flow cytometry based analysis, right? Be it FlowJo, FCS Express, all these different tools that are out there, and certainly all the cool things in R. We probably need to have more of an ecosystem in the single cell molecular cytometry space of tools. You know, there's oh gosh, Florian, help me out. What is the FlowJo for SickKids? Yeah. So you know, so the FlowJo folks have developed SickKids that, you know, helps us analyze the data, but we're still relying on informaticians to do this alignments and, you know, and those initial steps. So perhaps having out of the box tools to do that will help a lot with, you know, or to, you know, so that we're not reaching out, or maybe it's just a matter of us, you know, knowing enough R to be able to, you know, trigger those pipelines rather than having to wait a couple weeks for a bioinformatician to write us back, you know, in a busy institution. You know, I think we just need to have these have the tools that make this work a little bit faster in the single cell RNA space.
So I was just gonna, sorry, just quickly follow up on the point about the pre analysis pipeline. So we use the 7 Bridges pipeline that BD developed, and you know, now we're not really relying on anybody, right? So, you know, and of course we're trusting that that's a reasonable pipeline, but I think it must be, and we're getting data that makes sense. So I think there are, you know, starting to be ways where you can actually do the whole thing stem to stern if, on your own if you want.
Yeah, exactly, and that's one of the great things about the BD Rhapsody system. But for you know, is the 7 Bridges pipeline compatible with doing TEDx stuff? I don't know.
Yeah, well, yeah, I believe it is. Well, at least
I
think it is actually.
Well, 7 Bridges is just a provider of like
a pipeline
computing,
right?
of computing services.
Right.
So I think 10x is also moving to the cloud. I mean, in the past you needed to run it like on the command line, but I think they also moved now to a point and click cloud-based service, although I'm not entirely sure about that.
Mm-hmm.
I'd like to take a second to echo and give a +1 to what was said before about graduate students being a great resource. I think for, you know, when the people come to the core looking, you know, hear my data, there's I think the labs are really the place where the knowledge is going to live, I think, with the graduate students and the different PIs. I know we, at my institution, they've really organized a whole, it's been going on for years, where the graduate students have self-organized our workshops. They teach each other on how to do it, and they do it for flow, they do it for other technologies as well. And it seems to be working really well at my institution, that's where the knowledge resides, and that's where the work is being done, is with the graduate students themselves.
So I'll ask a question of follow-up. What happens at for you all at institutions where there are a lot of clinician investigators who don't have lab resources, who don't have graduate students, and who basically rely on the core for this stuff?
Okay, well, I'll take that one because we deal with that almost every day. It's a challenge, you know, and so on the one hand, we do have some capacity, although not nearly enough, to where we will do the staining. You know, so we'll do the wet lab work, we'll acquire it on whatever instrument, and we'll do the analysis. But we can't really do that much for large projects. We can do it for pilots and to help them, and then train people in their labs to do it. But it's a big gap, I think. And ideally, I think institutions, if they want to support that kind of sort of translational research, need to provide more resources to the platforms who can do it, to allow them, because it's challenging. I wish we could do more of it. We do some of it, but it's a challenge.
Yeah, I can imagine that would be a bit of a challenge. We don't see much of it in my facility, but definitely a lot of the similar concerns we have, even with smaller labs who don't have, you know, graduate students or people who are capable of doing the bench work, and it is tough to integrate. So we are getting near the end of our hour. So are there any questions that people in our audience would like to ask our panelists at this point? Feel free to put them in the chat box or unmute yourself and ask away. We'd love to hear from you, and I'm sure our panelists would too. While we wait for questions to come in, any final thoughts from our panelists on high dimensional cytometry in general, the future, things that you're particularly excited about? Silence.
I think it's kind of like the future is now. I know it seems like a really fluffy thing to say, but if you're not doing it, you will be. And if you don't know where to start, there's a lot of smart people we're all nice people and willing to help give pointers. There's lots of good reviews out there. If you don't know where to start on analysis, on protocols, cytometry is doing a really good thing with OMIPs. There's a lot of really good resources to get started. It all takes time, but I think it's going to behoove the people in SRLs to educate themselves. It's only makes you a better person.
I'll also plug in here, the Isac SRL committee has started an SRL SOP repository that you can link to on the Isac website. So it's also an excellent resource for SOPs, and we're looking for SOPs if anybody has any they would like to share there.
And as long as we're plugging things that are related to Isac, I think a large piece of this is networking, right? And being able to find resources and collaborate with people who can help us do more than we could do alone. And honestly, my whole career has been built on collaborating with people who could help me do more than I could do alone. And so one of the ways to do that is to become a leader in the Isac program. And so Isac itself has a leadership development program that's a great forum for networking. And there are 3 different tracks. There are tracks for people who are entrepreneurs. Those are the innovators. That's the innovator track. There is a track for people who are SRL affiliated, so people who are involved with core laboratories. That's the SRL Emerging Leader track. And then there's the Isac Scholar track, which is for academic researchers, for postdocs, graduate students, folks like that. And, I know Ryan, Florian, and I all are emeritus of that program and greatly appreciative of it. That program is actually, those programs are actually open for applications. And for those of you who are early stage investigators, I can't remember I think there was an age limit of 40, but that might not be the case anymore. I think there's a number of years, you just have to have not been in the field forever, not be a dinosaur, basically. We encourage you to apply. Where the applications process is open, and it's really a great cohort of folks to join and a great way to network and develop these resources.
100% agree. All the good things in my life have come from Isac, and just going to meetings. So if you don't know where to start, find some resources to get yourself to an Isac meeting. It's going to be in Quebec in another year is the CCMA, right? So it's not so hard to get yourself out there, and just the resources that are made available through these meetings is fantastic. The education that's done there, the meetings, you can expose the latest stuff. It's the best thing if you want to learn more, I think. Really good place to start. And the people that you meet there are awesome.
And on that note, since we're plugging stuff, we have one more session left in our SRL Perspective series. The last session is actually a big thank you to all of the vendors that have supported us throughout this whole yearly event. And so, yeah, it's our vendor showcase showdown, where everybody gets a couple of minutes to talk to us about all of the great new things that they're working on in development. Yeah, so please join us. We will definitely be having some prizes for drawing at the end, and some of these prizes include Amazon gift cards, $50 to spend on whatever you want. And also, one of the prizes is going to be registration to our next in-person core facility meeting. So this is going to be a core facility staff centered meeting, and it's going to be in the winter somewhere in some kind of resort, hopefully where we can hang out around a fire and chat with each other and connect again. So yeah, so if you can't make it to CYTO in Montreal, please do join us for our meeting in the winter. Yeah, we'll try to make it so that, you know, it's budget friendly and everybody who wants to can join us. Yeah, so part of that also is, if you guys followed us from the beginning, a lot of the vendor support that we're collecting is going towards a scholarship, I guess, of sorts for SRO people, and it's named after Chris Spring, who we miss very much. So yeah, this is going to be support from Chris to you, because we know that he will want you to go. So yeah, so these are all things that are in the pipeline. Hope you guys can join us, and hoping that Omicron is the beginning of the end and we will get to hang out and see each other again soon.
Dara, just briefly, there was one user question. Can we still answer that?
Yeah,
because it was
sure.
a very generic one.
Sorry about that.
No, that's fine. So I'll just read it to everyone. It says, would you please advise good resources to educate flow cytometrists to learn how to do sequencing? Are there a training data set with results to learn from bioinformatic tools? So my take on that is, I would point you toward 3 sources. The first one is both a paper and a bioconductor website that's been maintained by the Gotardo Lab. I think the title is called Orchestrating Single Cell Analysis in Bioconductor. It has been a 2020 paper, I think, in Nature Biotechnology. It's an excellent resource. If you go to the online portion of it, it's really every detail is there with references and what have you. If you want to have something that's more point and click, I mean, the platform that I know is SickKids from BD, where you can have like webinars and kind of play a little bit around with the data with point and click. And otherwise, I would go to the website. If you want to get into R yourself, I would also go to the website of the Sateecha Lab that has developed a package that is termed Surat, and they have like really, really good tutorials. And that's basically where I learned it. I mean, I learned it from there, and then just started playing with my own data and talking to people, and then iterative process, I guess. So those are the 3 resources that I would recommend that I feel are of high quality and where you can get going with the bioinformatics tools.
Yeah, and the vendors, in my experience, have been extremely responsive as well. So for the Rhapsody team has at least five field application scientists who specialize in AbSeq, and they're extremely knowledgeable. So the a lot of these tools that Florian brings up, these resources that Florian brings up, are sort of a little bit more on the bioinformatics side. But if there are questions about protocol, like how you set up your thermocycler and things like that, because many of us never saw a thermocycler until we left flow cytometry to start doing this stuff. That's an exaggeration, but they are very helpful in that regard as well. And I'm sure there are folks on the 10x side and on BioLegend side who will be fantastic resources as well.
All right, awesome. I think we'll wrap it up there for today. Thank you so much for a great discussion with all our panelists today. Thank you for joining us and giving your time for this hour. Thank you very much to our sponsors again. So BD is our series sponsor, BioLegend, Cytek, and Sony are the sponsors of this particular session. So thank you. And yeah, please join us for our last session of this series in March, and advertisements will be coming your way soon. Thanks so much and have a great day, everybody.
All right. Thank you. Bye-bye.
Bye.
Let's explore BioLegend's new flow cytometry resource guide. We have developed innovative reagents for flow cytometry for decades. This guide is packed with information on reagents for conventional and full spectrum flow cytometry, detailing characteristics of fluorophore families and dyes, including cell health and viability dyes. There's a section on buffers for sample prep, surface and intracellular staining, plus other flow reagents like our lyophilized human PBMCs and legend screen kits. Download your copy of the guide today.